use of adaptive optics retinal cameras in age-related macular degeneration. We are all aware of the current progress in therapies for AMD with promising results from many early phase trials and soon the first approval of a treatment for dry AMD. So in this very dynamic context, we propose to discuss the role and benefit of AO imaging. Over the next 30 minutes, we will focus not on science, but on personal insight with 3 clinical experts who very kindly agreed to share their experience and their vision. And now Marine is going to introduce our guests.
So hello everyone. For the roundtable discussion today, we're pleased to welcome Michel Paques. He is professor of ophthalmology at the 15 National Eye Hospital in Paris. He pioneered the development and use of adaptive optics retinal imaging in a wide number of diseases. Hello, Michel.
Hi.
Then we have also with us Andrew Lotery, who is professor of ophthalmology at University Southampton. Specialized in medical retina, one of his research interests is the molecular basis of AMD and developing treatments for it. Welcome, Andrew.
Thank you.
And our third guest today is Paulo-Eduardio Stanga, ophthalmology professor at University College London and director of the Retina Clinic London. Among other activities, he runs clinical trials on innovative therapies, including for AMD. So thank you, Paulo, for being with us today too.
Thank you very much, Marine.
So to start the discussion, I want to first ask Paulo, why did you equip your wonderful clinic with an adaptive optics camera? What are the needs that have driven you to acquire this technology?
Thank you very much, Marine and Nicolas, for this invitation. It's a privilege for me to join Andrew and Michel in this webinar. Well, I believe that adaptive optics imaging can contribute with new and complementary information to other ophthalmic imaging techniques such as ultrawide-field imaging, fundus, autofluorescence, and even high-resolution OCT. All of these technologies should form part of a comprehensive eye examination, in my opinion. The goal is to reach diagnosis as early as possible. Adaptive optics allows us to perhaps detect changes earlier than OCT and fundus autofluorescence. And this could be an ideal tool for screening and monitoring disease. Additionally, adaptive optics can assist us to analyze and measure microvascular structures with high precision, leading to better understanding of eye diseases, for example, in CSR, diabetes, or systemic hypertension. This is a tool that could perhaps allow easier reach of diagnosis when there are 2 differential diagnoses that are very similar. Let me share with you a case of CSR, central serous retinopathy. I would like to point your attention to a very recent publication from Bousquet, published in Retina this year, that showed in patients with unilateral CSR and the presence in the fellow eye of reduced density of foveal cones in the absence of decreased visual acuity and even photoreceptor line disruption on OCT scans. So let me share with you one of my patients. This is a 63-year-old male patient, type A personality, intermittently stressed, which first came to see me in March 2020. And we've been following him up with all sorts of imaging technologies, including ultrawide-field imaging, high-resolution OCT, and adaptive optics. And as you can see there is a reduced density of cones. There's an abnormal pattern of photoreceptor sorry, even in the resolution of fluid. So even though there is resolution of fluid, we see that there are permanent changes in the photoreceptors. Here you can see the changes between 11th of January and March this year, and you can see that there are persistent changes at the level of the photoreceptors. Perhaps this is secondary to a pachychoroid. You can see here high-resolution swept source, single-line wide field OCT scans. You can see the thicker choroid in the bottom image, the right eye showing a pachychoroid compared to the left eye. And we did not see any changes in the left eye, neither on OCT nor on adaptive optics. So we are using, for example, in this patient, adaptive optics to monitor the changes. This patient is not keen on any form of treatment. So we are trying to see if there is ongoing progression of these changes. So far, we've done adaptive optic imaging every 3 months with stable findings. So this is an everyday case where I wanted to show the benefits, perhaps, of adaptive optics outside age-related macular degeneration.
Thank you very much, Paulo. And now I want to ask Andrew about the fact that you recently implemented adaptive optics cameras in a very ambitious multi-center investigation in AMD. The project is called the Pinnacle. What is the purpose of using AO in this project?
Well, we're interested in understanding the natural history of intermediate AMD and why some patients progress quickly to late stages of AMD and some progress slower. So we really want to interrogate every layer of the retina, the RPE, the choroid, to really find the earliest changes that predict progression. And so adaptive optics gives us a tool to look at the cone photoreceptors, to look at the RPE, and just gives us a high resolution tool to investigate these patients and to see what novel phenotypes that we can pick up. For example, with looking at drusen with adaptive optics, and also to try to do a structure function correlation. So trying to see if the changes that we pick up in adaptive optics correlate with, say, microperimetry, and also with other multimodal imaging such as OCT images. So there really hasn't been that many prospective large cohort studies of intermediate AMD. This is one of the first, and it's adaptive optics is we think is a useful tool to look at high resolution and pick up the earliest changes that drive progression. And so we hope it's going to help us understand the etiology and, also, potentially identify structural biomarkers that could be used in clinical trials in the future as surrogate endpoints for late stage AMD, because AMD trials in geographic atrophy are difficult. They can take years to do. Progression can be slow. They're expensive to do. If we could come up with better phenotype patients, identify patients who could progress quicker or more uniformly, if we could have an endpoint before wet AMD or geographic atrophy, that might be helpful to do shorter, quicker trials and get us to treatments quicker. So adaptive optics is something that we're looking at to see if it can identify these early structural changes that would be useful in future clinical trials as endpoints.
Well, this is a very interesting perspectives. And now I want to ask Michel, because you and your team, you were real pioneers. You were the first to explore the geographic atrophy at the macroscopic scale with AO. Why did you start looking into it with adaptive optics?
Well, the question was to try to understand what's actually happening in this complex tissue modeling, such as seen in geographic atrophy. First, I forgot, of course. So, in recent years, work in histology in particular have been identifying the different various phenotype of cell involved in macular degeneration seen in AMD. And this is an illustration of the work from Curcio, showing the various phenotypes of pigment dispersion, of RPE cell dysmorphia, and so on. And this, I think, is an interesting biomarker that can be detected in vivo relatively easily, such as by OCT, for instance. On the left, you see an image of a drusen, which hyperreflected foci overlying it, and this makes a link with the previous presentation about intermediate AMD. This is a typical case of intermediate AMD. So we're interested in means of detecting this pigment, which may denote activity of the disease. And we've been doing this as an illustration of what we were expecting to see, and which we indeed see. It's over time. I think it's a time-lapse sequence taken over a few months. And at the summit of a drusen, we can actually see that the pigment is not static. It does not move. It's indicating that probably the presence of this pigment in the retina they're located into cells, probably in macrophages. So by documenting the change in the pigment disposition, we can actually also document the activity of the cell itself. So that appears as a very promising tool to understand some basic mechanism in dry AMD.
Well, thank you very much for all this very interesting insight into, I would say, what's needed to push forward the therapeutic approaches for AMD, and why you started to use adaptive optics in this context. So now let me ask you about like what you could see with adaptive optics, and also the vision on what this technology can bring in the future. So Michel, in Paris with your team, what have you learned so far with your investigations?
Well, we've been observing the particular, not only intermediate AMD, but also progression of atrophy, which was our first objective. And the idea was, can we measure more precisely the progression of geographic atrophy? And the response, non-surprisingly, the response is yes. And so this is an illustration of progression of atrophy. The atrophy on the right, the non-atrophic RPE on the left. And this was the first demonstration that AO can very precisely measure the progression of AMD over weeks, and let's say reasonably within months and sometimes within weeks, which I think addressing the issue of the duration of clinical trial that mentioned by Andrew is indeed maybe a game changer in order to appreciate more rapidly the effect or not of a given drug. But also we were interested in what happens to the other structure, such as a pigment, for instance. And the big surprise that was indeed there is also motion of these pigment, these dark spots that you see all over the place. So there are also during progression other processes that can be detected that we're actively studying currently. We recently published a paper about that, but not on adaptive optics, showing that what happened in the margins, that you can have a glimpse here on this sequence, also has importance to understand the progression of geographic atrophy. And I think that could indeed actually give big clues about not only the speed of progression, but also the actual mechanism of progression of the disease.
Okay, so you already replied to the question of where do you see the best applications of the findings. So is there anything you want to add?
Well, I could mention that it's I mentioned the histology data at the beginning. I think it's what we see by adaptive optics has so much detail that we need to go back often to histology to understand what we're actually seeing. And this also opened a new field of the observation of the retina. For instance, on the atrophic side, on the right side of the image, you see all these dots which resemble cone mosaic, which are not cone mosaic. In fact, it's something maybe related to Bruch's membrane itself, maybe BLAMD. And that's why we need to also to cooperate with histologists to understand exactly what could be the origin of this mosaic. And also tell us that what you see in the fundus as a mosaic is not necessarily a photoreceptor mosaic. So it challenges many of the interpretation of AO images that we've been doing for years. So it gives some new clues, but also raises additional questions. And this may also, having the answers to this question will be helpful to understand the features of adaptive optics imaging in other diseases such as PRSC, as mentioned by the first speaker, for instance.
Okay, and Andrew, in Pinnacle project, what have you observed so far with adaptive optics?
So it's been interesting hearing from the other speakers. So we've seen different phenotypes of drusen, quite striking differences. Here we're just showing an example of cuticular drusen, where there's sort of amorphous material, which has a very different appearance to subretinal drusenoid deposits. So we're seeing we're seeing distinct phenotypes of drusen with adaptive optics phenotypes. I was also interested, we've also seen that when you can't see the interdigitation zone of the RPE cells, you don't get a bilayer of the RPE, it's more difficult to see the cone cells and the cone mosaic. And I was just struck by the CSCR image that Paulo showed, that it wasn't a high resolution OCT scan, but the lack of the cone mosaic is something that we see when we can't see the RPE structure in good detail and in the Pinnacle study. So there, and we've also seen that pigment over drusen that Michel has mentioned, and we I think based on his work, we did wonder if those were macrophages that we were seeing, and we're seeing this change over time. And it perhaps highlights, something that we don't think about a lot with OCT is, perhaps there's macrophages crawling all over drusen in intermediate AMD. It's early exploratory findings, and I totally agree. We need to understand more what we're looking at. But just thinking of potential targets for treatment, maybe some sort of anti-macrophage treatments, is if we were more aware of it, if we confirm those findings, that might be a therapeutic approach that hasn't really been thought of so much with the drive to anti-complement therapy. So I think it's giving us new ideas for etiology, and just as Michel said about defining new endpoints so that we can do more targeted trials and quicker trials, and it's potentially, it could be very useful for that. So we're still very much amassing the data and trying to understand what it means, but we are seeing some interesting things similar to what Michel said, with pigmentation over drusen, different drusen phenotypes, and then alterations in the RPE, which makes the cone mosaic more difficult to see as well, which is interesting. So there's lots of data that still needs analyzed, but it's stuff that we can't see with other modalities, which is interesting.
And you mentioned earlier that one of the aims of the project is to try to see if you can identify earlier patients who would progress to late stage. What would it mean to be able to identify those?
Yes, so, one thing, you could stratify patients either for their clinical follow-up. You might want to see patients you're predicting are going to progress quickly sooner in your regular clinics. You might want to see patients who progress faster. They might be a cohort of patients you might want to have in a clinical trial, because you'd get to an endpoint quicker. So it might help us create a personalized prediction for patients about how long what the course of their disease is going to be like, and that could be useful both for trying to detect early progression to disease that needs treatment, and also for selecting patients for clinical trials. So I think trying to build a better prediction of progression, and we're trying to build that with trying to find the most relevant biomarkers, if you like, that could feed into a personal prediction model. And then one of the ambitious goals of the project is actually to create a personal prediction model for patients. So a lot of work still to do, but we're getting lots of interesting data.
So thank you. Now we have still one question to ask to Paulo Stanga. So you plan to use adaptive optics imaging in several clinical trials. How do you think it will improve the evaluation of retinal therapies?
Well, thank you for the question, Marine. We are in discussion with several of the companies that are setting up trials, and because we think that, echoing some of what the other two speakers said, adaptive optics should ideally be used in clinical trials for participant selection, stratification of disease, and monitoring treatment safety and efficacy. We need to be able to identify earlier biomarkers of both progression and regression, so that these will allow us to evaluate better the therapies we are looking at. There's no doubt, and this is a constant discussion at meetings nowadays, especially gene therapy meetings, that we need new endpoints in clinical trials. Visual acuity is not ideal. So in clinical trials of IRDs, inherited retinal dystrophies, and AMD, we do need new endpoints. So we are looking at the moment at fundus autofluorescence, as we know, but once we see changes in autofluorescence, we are already very late in the race. There are already established changes that it's most likely we are not going to be able to turn around, as we can see in this patient of ours with geographic atrophy. Hence, the need to look at adaptive optics as part of clinical trials. We've been also looking at the ellipsoid layer on high resolution OCT. Again, we need to look at earlier biomarkers of progression. We're through an era of gene therapy and complement modulation therapy. And we need to not look at fundus autofluorescence alone. We need to complement this with adaptive optics. We need to look at nascent GA, iRORA, and not cRORA. And all these future studies, or the new studies coming up in using these therapies, we need, ideally, should include adaptive optics. There's no doubt that we all need to look at the work from Drs. Gocho and Paques. They are pioneers in this field, so it's a privilege to be sharing this webinar with them. These are some of our results, and as you can see, if you advance the slides, Marine, please. Here we are imaging area of GA, and on the margin of the area of GA, as Michel pointed out before, it's essential. We need to know what's happening in the periphery of the area of GA, a little bit like what happens in diabetics if we look at the peripheral retina. So we can see all these white and black dots and the position of pigment. We can even see a choroidal vessel through the area of GA and the loss of photoreceptors. But there's no doubts that in the opinion of myself and others, that adaptive optics really has a role. And we need to learn more about what we are seeing, and of course collaborate with histopathologists to have a better correlation between what we see, what those changes in pigmentation, changes in the white and black spots, drusen, and changes in the morphology of cellular mosaics really mean.
So thank you, Paulo, Michel, and Andrew, for sharing with us your insights and vision today.
I have a question from the Q&A.
Yes.
Is it possible to predict? Is it possible to predict AMD activity from AO imaging? So Michel, do you want to take it?
Yeah, that's a crucial question that, it depends on if you have or not follow-up images. If you're talking about a single image, which is, let's say, the daily problem of these patients, we expect that there is a correspondence between the number of pigment spots that we see and the presence of inflammatory cells, because histology done in particular here in Paris show that most of the pigment spot, the melanin spot that you can see dispersed in GA cases are located into macrophages. So it seems logical to think that more pigment there is, the higher the activity is. This remains to be proven. It's not yet demonstrated clearly. My impression that it's probably far more complex than just that. So it's an open question, but again, given the fact that I think adaptive optics can bring information about what happened in the margins, then we may end up with novel biomarker that may be more provide hard data on the individual risk of progression, and especially progression toward the fovea, which is the main question that patients are concerned with.
Thank you, Michel. So now it's time to wrap up. Meanwhile, I want to say a big thank you to our guests. It's very nice from you to support this webinar. And thank you everyone for your participation.
Thank you.
So goodbye, everyone.
Thank you, everyone.
Thank you.
Thank you.
Bye.
Goodbye. Thank you very much.